afternoon, children. Yesterday, we discussed the principles of inheritance and variations, objective questions. Then now we have to discuss the remaining questions. This is the most important topic for NEET point of view. Here, second year syllabus, there are 16 chapters are there relevant with NEET. Of these, the most important chapters are genetics, molecular basis of inheritance, principles of inheritance and variation, followed by reproduction, sexual reproduction in flowering plants, human reproduction. So here, this is the topmost priority, genetics. Now here, let us discuss the remaining questions. Which of the following cannot be detected in a developing fetus by amniocentesis? Question number 16. Which of the following cannot be detected in a developing fetus by amniocentesis? Option A, Down syndrome. Option B, jaundice. Option C, Klinefelter syndrome. Option D, sex of the fetus. You see, question number 16. Which of the following cannot be detected? Following cannot be detected in a developing fetus. Developing fetus by amniocentesis. Option A, Down syndrome. Option B, jaundice. Option C, Klinefelter syndrome. Felter syndrome. Option D, sex of the fetus. Here, this is the question. Here, which of the following cannot be detected in a developing fetus by amniocentesis? Amniocentesis means amniotic fluid. This amniotic fluid, it has been taken and analyzed by using the cell biology techniques. On analysis of amniotic fluid, what we have to determine here? Genetical disorders. Amniocentesis techniques, this is used for detection of genetic disorders. Next one, second thing, this is used for detection of sex of baby. Used for detection of sex of baby. For this case, amniocentesis has been used. Amniocentesis technique, it has been used for detection of genetic disorders, sex of baby. So that you see Down syndrome. Down syndrome means, what is the Down syndrome here? It is a genetic disorder. Why then genetic disorder, Down syndrome here? Down syndrome, autosomes will be there. Then what is the Down syndrome? Extra chromosome has been added. Here, one extra chromosome has been added in the Down syndrome. Autosomes are added with one extra chromosome, plus here XY is there. 21st pair has been added with one extra chromosome. So addition of one extra chromosome for this 21st chromosome, that can be called as the Down syndrome. So that here in this case 47 chromosomes will come, but here by means of amniocentesis, it is a chromosomal disorder. Chromosomal disorder means here this is relevant to genes. On chromosome, what should be situated? Genes. So chromosomal abnormalities, genetic abnormalities, as well as determination of sex of baby, this technique has been used. So it is a chromosomal aberration. Henceforth, here we can identify by using amniocentesis technique. Jaundice, what is the jaundice here? This is bile juice, liver defect here. This is here. If the bile juice it is added to the blood, by the leakage of bile juice into the blood. Blood has been what have diluted with the bile juice. Henceforth, your eyes, then your nails turn to the yellow color. This is a liver disorder, not this chromosomal. Henceforth, your jaundice would not be detected by this here amniocentesis technique. Next, you see Klinefelter syndrome. What is meant by Klinefelter syndrome? It is relevant with the male. In this male, it is a chromosomal aberration. Chromosomal aberration means how many chromosomes will be there here? Here is also 47 chromosomes. Down syndrome 47, here 47. But it is autosomal trisomy. This is allosomal trisomy. Allosomal trisomy means one extra chromosome added to sex chromosome. What is the extra chromosome? X chromosome. You see this here, Down syndrome means 21st pair is extra. But here, when you observe this Klinefelter syndrome, autosomes, what is the Klinefelter syndrome? Here, when you observe this Klinefelter, autosomes as is 44. But its sex chromosomes X, Y. But extra what is added? X is added. One extra X chromosome is added, that is Klinefelter syndrome. There is how many chromosomes will be there in Klinefelter? 47 chromosomes are there. But extra chromosome, where it is added? To the sex chromosome. So here Klinefelter syndrome, chromosomal aberration, trisomy. So that it also detected by amniocentesis technique. The sex of the baby, whether male or female, how it is decided here? By means of chromosome. Generally, if you see the sex of baby, autosomes plus XX is there. How it will be decided? This is a female. Autosomes plus XX. This is a female individual. Autosomes plus XY. Then it will be called a male. This is a male individual. So on the basis of chromosome configuration, we can determine the sex of baby. But amniocentesis used for genital abnormalities, chromosomal disorders, as well as used for detection of sex of baby. For determination of genetic disorder, as well as what purpose using chromosomal aberrations. What is meant by chromosomal aberration? Change in the chromosome number. Maybe addition of X chromosome or loss of chromosome. Extra chromosome may be added, aberration. Or from existing chromosome may be lost, aberration. Maybe a 2n plus 1, or 2n plus 2, or 2n minus 1, or 2n minus 1 minus 1. Like this here, chromosomal aberrations are there. So chromosomal aberration, genetic disorders could be analyzed by amniocentesis technique. As well as this is helping for determination of sex of baby. Henceforth, what is the answer? Jaundice, it is relevant with the liver. Therefore, here amniocentesis is not using for the detection of jaundice. Remaining Down syndrome, Klinefelter syndrome, sex of the fetus, they can be determined by the amniocentesis technique. Henceforth, the answer is what is here. Jaundice is the correct answer. Then we can see this next answer. If both parents are, this is the 17 questions. You see, already you are served with the papers, question papers here. So what the question bank here? We can see the questions and see this here. If both parents are carriers for thalassemia, which is an autosomal recessive disorder, what are the chances of pregnancy resulting in an affected child? The question you see, 17th question. If both parents are carriers, both parents are carriers for thalassemia. Both parents are carriers for thalassemia, which is an autosomal recessive disorder, which is an autosomal recessive disorder. It is an autosomal recessive disorder. What are the chances of pregnancy what are the pregnancy resulting in an affected child. Here, option A, 25%. Option B, 100%. Option C, no chance. Option D, 50%. Given question, thalassemia. Thalassemia, sickle cell anemia, autosomal recessive disorder. This is what is this disorder? Autosomal recessive disorder. Autosomal recessive disorder means what is the gene configuration? Autosomal means this is applicable for both. It is whom it is applicable for both male and female. Male and female in same pattern. Here there is no change here, same pattern. The pattern of inheritance of autosomal disorders, they are common for both male and female. This is not a X and Y linked. It is not sex chromosomal disorder. In given question, what they mentioned, autosomes. Autosomes pattern is inheritance patterns of autosome. This is same in both male and female. Therefore, you see it is a recessive disorder. Recessive disorder means the alleles of a gene both should be in homozygous condition. Then here disease is happening. Suppose you see this is a AA gene. This is the dominant allele. Two dominant alleles are there. Thalassemia, it would not occur. Whether it is male or female or whatever it may be, this is no effect. No effect it is. Suppose the alleles of a gene, dominant allele, recessive allele. Capital letters means dominant allele. Whatever I am writing, this is A means it is a allele. This allele is a capital letters allele is a dominant allele. Small letter allele is a what is this here? Recessive allele. So you see both dominant alleles, homozygous dominate, no effect. This is heterozygous dominate, no effect. How this is called here? This is called as a homozygous dominate. Like this genotype, it would be called as a homozygous dominant. It is a homozygous dominant. And here this is called as a heterozygous dominant. How this is called heterozygous dominant. Now you see like this is there. What is this here? Homozygous recessive. Small a alleles. How they can be called here? Homozygous recessive. This is the homozygous recessive. In this gene pattern or allelic pattern, what will be their effect? The individual is affected. In the thalassemia, suppose A allele is responsible for thalassemia. If the alleles organized into two dominant alleles, thalassemia could not occur. If it is one dominant, one recessive, thalassemia could not occur. If these alleles are, what would be there? Both of these here recessive pattern. That means here small letters are recessive pattern. Then in that case, disease would happen. So what is the case here? Both parents are carriers. Carriers means this is no effect, this is effect. But carrier means one dominant allele, one recessive allele. At that case, how they can be called? These heterozygous individuals, no effect, but they can be called as the carriers. The heterozygous configuration can be called as the carriers. In case of autosomal recessive disorder, one dominant allele, one recessive allele, it is there. At that case, they will be considered as the carriers. So here, what is the question? Both parents are carriers. So here, male is carrier, female is carrier. In between meeting is happened. What is the chances of progeny to have a thalassemia? So we can make a cross. Here, in this case, we can make a cross. What is the cross? You see, this is a carrier genotype. Here, thalassemia carrier into thalassemia carrier. In this, one is the male parent, another one is the female parent. From male parent, how many types of
Sperms will come. Heterozygous condition means here two types of sperm. One 50% sperms they have a dominant allele. 50% sperms they have a recessive allele. Then in this case, what is ova? 50% ova they have a dominant allele. 50% ova they have a recessive allele. Now you see this dominant allele sperm can fuse with this ova. Then now individual has been born. This individual configuration capital A, capital A. Next one, this one is mating happens. What is the configuration? Capital A, small a. Suppose here mating is happens here. What is this here? Capital A, small a. Suppose this sperm is fusing this ova. What is the configuration? Small a, small a. Carrier male parent, carrier female parent. If these two are crossed, what is the chances to have a thalassemia? Now you see, this is the possibility of sperms, this is the possibilities of ova. Then here combinations, after possibility combination. What is the combination? This sperm may mate with this ova. Next case may be mate with this ova. If it is fused this ova, what is the configuration? Capital A, capital A. Already I told homozygous dominant means how they can be called here? Normal individual. And here next case, what is this here? If this one fused with this one, how it will be called? Heterozygous dominant. Heterozygous dominant means they are normal, but how they can be called? They can be considered as a carriers. These are also how they can be considered. They are normal, but how they can be considered? They are the carriers. Last choice here, two alleles are recessive. At that case, how it will be affected? These are the affected individuals. So, how many individuals are there total? One, first individual, second individual, third individual, fourth. Out of four individuals, how many are affected? One individual is affected. So here, what is this probability? 1 by 4. 1 by 4 means probability is 1 by 4. Percentage, what is the percentage? Here you can 1 by 4 into 100. Then 100 by 4. 100 by 4 means what is the percentage here? 25%. So what is the given question? Affected. Percentages are given. So what is the percentage is affected by thalassemia? Answer is A, 25%. If in thalassemia, how it is happens, if a cross is made between two carriers, what would be the probability or what is the chances to have a thalassemia in their crossing? 25% progeny or 25% children, they have been affected with the thalassemia. It is a autosomal recessive disorder. Same inheritance pattern applicable for the sickle cell anemia. You have a syllabus here, sickle cell anemia is there, thalassemia is there. And both cases here, there is a choice to ask the question. In place of thalassemia, if they give this about sickle cell anemia, you follow this here, same procedure, same pattern. So this is question number 17 over. Next we go for question number 18. You see, question number 18. If two persons with AB blood group marry and have sufficiently large number of children, these children could be classified as A blood group, AB blood group, B blood group in 1:2:1 ratio. Modern techniques of protein electrophoresis reveal the presence of both A and B type proteins in AB blood group individual. This is an example of. Here, what is the question here? Once you see two persons with AB blood group married and have sufficiently large number of children, these children could be classified as A blood group, AB blood group, B blood group in 1:2:1 proportion. Then modern techniques of protein electrophoresis reveal the presence of both A and B type of protein in AB blood group individual. This is an example of. That means the expression of two alleles at heterozygous condition is called as which phenomenon? Codominance. What is the actions they have given? partial dominance, complete dominance, co-dominance, incomplete dominance. But it is relevant with this. What is this answer? Co-dominance. So you have to see, note down this. If two persons with the blood group, if two persons AB blood group marry and have sufficiently large number of children. These children could be classified as A blood group, AB blood group, A blood group, AB blood group, B blood group, in 1:2:1 ratio. Modern electrophoresis proteins reveals presence of both A and B type proteins. B type proteins in AB blood group individuals. AB blood group individuals. This is an example of partial dominance, complete dominance, co-dominance, incomplete dominance. You see this here question, two individuals. What are those two individuals? AB blood group individual. Here, this is the genotypes of AB blood group individual, IA, IB. Man is AB, woman is also AB, like this. What is the, suppose this genotype is man, this genotype is woman. This is the man genotype, woman genotype. From man, what will come? Spells. 50% spells what they have? IA. 50% spells what they have? IB. From this, what is the configuration? You see 50% of over IA, 50% of over IB. Now you have to see, suppose A with A, what will come? IA, IA. This is, who is these children? A blood group children. This is the question only. How the question they have given, if a marriage is held between AB, then AB into AB. One AB is man, another AB is woman. They are having children. What type of children are there? A group children, B group children, AB group children. Why is AB blood group is there? Both type of antigens present here. This is an example of what phenomenon they are asking. Simple answer is here, the expression of both alleles at heterozygous condition, that is called codominance. Normally, at heterozygous condition, one allele can be expressed according to what is the law of dominance. Mendel proposed the law of dominance, but it is abnormality, deviation from Mendel dominance law. You see, this is an A group. Next year, what will come here? This sperm, suppose it is mating. What will come? IA, IB, LL. This is a question they asked. Here we are reading not A, not B. What we are reading? AB. Why AB reading here? Both are equally giving priority. A is expressing, B is expressing. Both alleles are expressed at a time means codominance. So AB blood group is example. These children, what they have? AB blood group. Next you see B. What is this here? AB blood group. This is the AB blood group. Next, finally, what is coming here? B blood group. What is the ratio they have come? First one is A blood group. IA. Out of 4 children, how many A groups are there? 1%. Out of 4 children, how many AB groups are there? 2% here, 2 individuals. Out of 4 individuals, how many having B group here? 1 individual. So what is the ratio? It is AB, B, 1:2:1. This is the question what they have given. The children, they are showing the proportion 1:2:1. If a marriage is held between AB into AB, the resultant children, they have A group, AB group, B group in 1:2:1. By means of electrophoresis of protein of AB group, these both are equally expressing. Why? What is the reason? Partial dominance, not partial dominance. Partial dominance means it is a incomplete dominance. What is meant by partial? Partial and incomplete, same only here. One allele is, suppose you see here A is allele, dominant allele, recessive allele. But here otherwise A, B. Suppose here 2 different alleles you take, if there is any confusion. But what is expressing? Only A is expressing, B cannot be expressed. So that out of 2 allele, only what is this? 1 allele is expressing, another is not expressing. Which would not be expressed, now it will be called recessive. Which is expressing, now it will be called dominant. Therefore, here you see what would be there here. Here the priority is given. We are not reading A, we are not reading B, we are reading AB, giving priority for, because both proteins expressing, both genes expressing. Therefore, it is called codominance. At heterozygous condition, the expression of 2 different alleles equally, that phenomenon is called as the codominance. What is example for codominance here? AB blood group is example for codominance here. So here the answer what is here? Codominance is correct. Complete dominance means law of dominance here. That is. So here what is here? Complete dominance here, law of dominance here. This is a law of dominance. Law of dominance here explained by the Mendel. Incomplete dominance means Mirabilis jalapa. In 4 o'clock plant here, Snapdragon or 4 o'clock plant, we have seen incomplete dominance. One dominant parent, one recessive parent. Both are crossed. Then what would be there here? Neither dominant parent, neither recessive parent. Neither dominant or recessive, both parent characters could not be seen. Any one parent character could not be seen. That can be called incomplete dominance. If red color flower, pink color flower, red color flower, white color flower, these 2 are crossed. What has come? White has come. So that is incomplete dominance it would be. So that here incomplete dominance, partial dominance, complete dominance, they are not explaining the AB group. What has been explained? Codominance explaining the AB group. So answer, what is the answer? Codominance is the correct one. This is wrong, this is wrong, this is wrong. The correct answer is the codominance. This is the element with the even blood group 18%. Now we go for another one, 19th one. Which idea is depicted by a cross? Idea is depicted by a cross. In which the F1 generation resembles both parents. Both parents. This inheritance of one gene, inheritance of one gene, inheritance of codominance, incomplete dominance, incomplete dominance, complete dominance, complete dominance. They are already, you know about incomplete dominance, complete dominance, codominance here, these phenomena. But this question relevant with the previous question only. The expression of both alleles equally, both parent characters are expressed. How it will be called codominance. Codominance is nothing but the expression of both alleles or both parent characters equally. That phenomenon is called as the codominance. Here, which idea is depicted by a cross in which here both parent characters are seen in F1. If both of the parent characters are seen, codominance. Here, next incomplete dominance means neither of the parent characters seen. Here, complete dominance means what is the complete dominance? Any one of the parent characters, either of the parent characters. Suppose parent A and B is there, but C comes instead of A or B. If C character will come, that it will be called here incomplete dominance. Parent A, parent B, cross. There is no A parent character, B parent character. New character is coming, incomplete dominance. If the cross is A into B, both parent characters A, B is coming, then it will be called here codominance. This is codominance. Complete dominance means suppose here A into B. Here suppose it is dominant character, it is recessive. Here A, dominant parent character is C. That is how it will be called here law of dominance. This is dominant parent, recessive parent. In F1 generation, dominant parent character is seen. That it will be called here law of dominance. This is the law of dominance. This is the codominance. This is what is this phenomenon? Incomplete dominance. Incomplete dominance. What is meant by incomplete dominance here? If a parent's dominant and recessive parent characters are here, two parents are crossed. But here what would be their new character is seen? Neither of the parents, dominant or recessive, neither of the parent characters are seen. That is how it will be called incomplete. Codominant means both characters, both parent characters are seen. If a cross is made between two individuals, both characters are seen, codominance. Here, if cross is made between two parents, dominant parent characters are seen, law of dominance. Here, our question, both parent characters are seen, so that answer what is this? Codominance. This is not relevant, this is not relevant here. But what is meant by inheritance of one gene? Monohybrid cross. Monohybrid cross here, it is not giving this codominance. What is giving? Law of dominance. Here, in the inheritance of one gene, it is following what pattern here? Law of dominance pattern is following. This is following the law of dominance. So that here, codominance pattern is not followed by the inheritance of one gene. Inheritance of one gene is nothing but what would be there here? This is called monohybrid cross. Inheritance of one gene, otherwise it is also called as the monohybrid cross. So according to given question, the answer is the codominance. Next one, question number 20. Which one is the incorrect statement with regard to the importance of pedigree analysis? It confirms that DNA is a carrier of genetic information. It helps to understand whether the trait in question is dominant or recessive. It confirms that the trait is linked to one of the autosome. It helps to trace the inheritance of specific trait. Here I will discuss here. I can write on the board. Then. Which one is incorrect statement? with regard to the importance of pedigree analysis. Which one is incorrect? Here, it confirms that DNA is the carrier of genetic information. It confirms that DNA is the carrier of genetic information. Here, it helps to understand whether the trait in question is dominant or recessive. It helps to understand whether the trait in a question is dominant or recessive. It confirms that trait is linked to one of the autosomes. It confirms that trait is linked to one of the autosomes. It helps to trace the inheritance of inheritance of specific trait. Here, yesterday we discussed this pedigree analysis. Generally, pedigree analysis means this is a family tree. Family tree means you see here. Yesterday we have drawn one diagram. This is there here, like this is here, like this. Here now some children are born. They are having this here defect. Next year no defect. Again defect has been there. Suppose here these are married to normal one. Now here there is a what would be the normal children are born. Again it's normal to normal. What will come? Defect happens here. Defect is there, and here no defect. This is pedigree chart. Suppose here you see defected male, normal female married. Then what these children? These are the affected son, normal son, affected daughter. Again, this affected daughter married to normal son. All are the normal. Again, this normal to normal. What came here? Affected as. Why? Yesterday we discussed this recessive disorder. What type of recessive disorder? Autosomal recessive disorder. This is the autosomal. So pedigree analysis means the graphical representation of the family history. In this family history, they are revealing the diseases. If the disease is the dominant or recessive. According to inheritance pattern, this disease, it may be dominant disease or recessive disease. Whether they have disease or not. This family history, it would be decided by this one. So it is helping for what would be there. It is helps to understand this here disease. In given question, whether they have the disease or not. But given disease is dominant or recessive, it helps. And here, whether it is autosomal disease or allosomal disease, if relevant with the autosomes, concerned gene is relevant with the autosomes or sex chromosomes, it would be understood by pedigree analysis. And it also here inheritance of specific trait. It is a sickle cell anemia, otherwise it is a hemophilia, color blindness, specific trait would be studied by using pedigree analysis. But what would not be studied? DNA, it confirms that DNA is a carrier of genetic information. This pedigree, it is not proving this DNA is a carrier for genetic information. Just it is revealing this here disease history of family tree. Graphical representation of the disease history of family tree from over the several generations can be called pedigree analysis. But in these diseases, whether they are autosomal or sex-linked, whether the disease dominant or recessive, those would be studied by pedigree. But these three actions revealing with pedigree chart. First action is not relevant with the pedigree chart. Henceforth, what is the answer? This is the answer is A. We need incorrect statement. B, C, D, three statements correct to the given question. A action is the incorrect to the given question. Therefore, the answer is, what is the answer? A is the answer. This is the answer. Now, next question. Down syndrome in human is due to come on. Three X chromosomes. Three X chromosomes. Three copies of chromosome 21. Chromosome 21. Monosomy. Two Y chromosomes. Two Y chromosomes. You see Down syndrome. Down syndrome means, what is the Down syndrome? This is human chromosome, human cell. In this here, first pair, second pair, third pair, fourth pair, fifth pair, sixth pair, seventh pair, eighth pair, ninth pair, 10th pair, 11th pair, 12th pair, 13th pair, 14th pair, 15th pair, 16th pair, 17th pair, 18th pair, here 19th pair, 28th pair, 21st, 22nd pair. This is here 22nd pair, 23rd pair. This is. It is the 23 pairs of chromosomes are there. One is X chromosome, Y chromosome. It is 22nd. This is 21st chromosome. What is meant by Down syndrome? The addition of one extra chromosome to the 21st pair. So in 21st pair, how many chromosomes are there in 21st chromosome? Instead of 3 chromosomes. Each set you see 2 chromosomes. Each set you see is 2 chromosomes. Normally diploid means normal cell. How do you have to write here? Normal human cell. Normal human cell 2n equal to 46. N equal to 23. 23 sets are there. Each set 2 chromosomes are there. But what is meant by Down syndrome? Abnormality. Abnormality means 2n plus 1. Down syndrome means, what is the Down syndrome? 2n plus 1. 2n plus 1 means how many? 47 chromosomes. That means one extra chromosome is added. Where it is added? To this here 21st chromosome set. In the 21st chromosome set, total 3 chromosomes. By one extra chromosome is added. So here 3 X chromosomes not. 3 X chromosomes means what should be there? 23rd pair is disturbed, triple X syndrome. That is not linked to this here. What is this here? Down syndrome. Super female triple X here, not having this Down syndrome. And 3 copies of 21st chromosome, yes, 3 copies are there. Monosomy means 2n minus 1. What is monosomy? From diploid set here, one chromosome is removed. Therefore, how many chromosomes will come? 45. But monosomy example, here, what is the example for monosomy? Turner syndrome. Example for this here, what is the monosomy example? Turner syndrome is example for this here, monosomy. So that it is not relevant. 2 Y chromosomes, what is this syndrome? Jacob syndrome. Here 2 Y chromosomes, Jacob syndrome. This is Jacob syndrome. It is Turner syndrome. It is Down syndrome. 3 copies are there. So relevant answer, what is this here? B. Triple X super female, extra X chromosome is there. Therefore, here what is the answer? 3 copies of chromosome 21. In chromosome 21, 3 copies are there. That is called this here. What is this here? Down syndrome. It is the autosomal trisomy. Therefore, the answer is the, what is the answer? B is the answer. A wrong, C wrong, D wrong, B is the relevant one. So answer B. And 22nd one. A normal vision man whose father was colorblind marries a woman whose father was also colorblind. They have their first child as a daughter. What are the chances that child would be colorblind? You see this here, I am writing this question. A normal vision man, normal vision man, whose father was colorblind, whose father was colorblind, marries a woman, marries a woman whose father was also colorblind. They have their first child as a daughter. They have their first child as a daughter. What are the chances that child would be colorblind? What are the chances that child would be colorblind? What are the chances that child would be colorblind? Here, option A, here 100%. Option B, 0%. Option C, 25%. Option D, 50%. Like this here, 4 options there were. So you see a normal vision man. You see this is a XY man. Normal vision means XY chromosomes are there. They are whose father? What is his history? He man means he born from father and mother. His father history, what is this here? Whose father was colorblind? Here, a man, normal man. This man is, what is his history? Normal man. Normal man, father is colorblind. Normal man, father, what is his colorblind? But father is colorblind means the son colorblind or normal? Normal only. How normal? This color blindness relevant with this, what is this here? X-linked chromosome, this defective gene where it is present on X chromosome. So X chromosome whom it is passing? To daughter, not son. If father have color blindness, son there is no color blind. Why? This color blindness, it is relevant with the X-linked. X chromosome inherit to the daughters, not son. So here, X-linked disorders, if father is problem means there would be no problem to son. So here this man is normal only. This man whom he married this year, this is married. Here, where it is the one woman, she is a woman. But what is the woman configuration? XX. Colorblind woman, whose father was also colorblind. Now here, this father is there. How she was born? Born from parents. What is his parents here? Father and mother. But what about this father history? Father history is the colorblind. So this colorblind gene passed to women? Yes. Here you see, he is a man, she is a woman. But this man father was colorblind, but he is normal. Why normal he is? Y is normal because of here from father he got Y chromosome. Male children they got Y chromosome from father. Female children they got X chromosome from father. So here father is affected. This affective gene where it is coming? Woman. But only one X chromosome is affected. No problem here. Problematic gene is there, but problem could not express. It is autosomal recessive. On both X's, defective gene is there. Character can be expressed. Now this history. Now they are married, but here they got a daughter. In this daughter, what is the choice to have a color blind? Now you have to see how many types of sperms will come. Two types of sperm. X chromosome sperm and here next to Y chromosome sperm. Two sperms are there. And now this is here this ova defective gene containing X chromosome and normal X chromosome. Now you see what it is here. This X chromosome sperm is mating. What it is X and X defective gene. These are carriers. These are the carriers. Next case, what is the daughters here? XX, like this. Next, what is the choice here? Now you see X, Y. Here this son is affected. Son is affected. Next, what is this here? Normal son. 4 offsprings are possible. But first daughter, carrier daughter, normal daughter. Next one here, affected son, normal son. But what is the question? Daughters. What would be the probability of affection of this color blindness in daughters? Not children, they asked. Children asked 1/4, 25%. They asked daughter means out of these two daughters here, no one affected. And so what is the percentage to have a color blind in daughters here? 0%. Daughters, no daughter is affected. So answer what is here? 0%. Out of 4, no one is affected here. Therefore, what happens here? 0/4 means what is here? 0% it is. So that here, what is here? No one is affected here. 0% here. So you see this here. This answer is the 0%. It is the what is this color blindness inheritance pattern. Next question. 23. F1 generation in Mendelian cross. F2 generation. In Mendelian cross, showed that both genotypes and phenotype ratios. Genotype and phenotypic ratios. The same as 1:2:1. It represents a case of. It represents a case of. What it is representing here? Codominance. Codominance. Hybrid cross. Hybrid cross. C. Monohybrid cross with complete dominance. Cross with complete dominance. Monohybrid cross with incomplete dominance. with incompleted dominance. You see this here, F2 generation in Mendelian cross showed that both genotypes and phenotypic ratio 1:2:1. You see here phenotypic ratio, genotypic ratio. Both are the same. Suppose law of dominance, you know. Law of dominance, what is the ratio? Phenotypic ratio 3:1, genotypic ratio 1:2:1. So that what happens here, complete dominance not applicable. Dihybrid cross. Dihybrid cross, what is the ratio? Phenotypic ratio 9:3:1, genotypic ratio 1:2:3:4:1:1:2:1:2:1. Therefore, here dihybrid cross ratios not equal. And codominance. What is the codominance here ratios? Codominance AB blood groups here. But phenotypic, genotypic same. Phenotypic ratio, genotypic ratio. But it is a Mendelian cross. But codominance first two, that is not a Mendelian cross. Maybe here it will come 1:2:1, but it is not non-Mendelian. Henceforth here not following Mendelian. But here what is following here? This is monohybrid cross with incomplete dominance. Here incomplete dominance means you see what is the incomplete dominance here? Mirabilis jalapa we can see. Here Mirabilis jalapa means suppose red color flower Mirabilis jalapa. This is a red flower Mirabilis jalapa. Crossed with white color Mirabilis jalapa. This is recessive. From this, what the gamete, capital R, small r. These two are crossing. What will come? Capital R, small r will come. Then this is F1 generation. We go for F2. F2 means what is the F2? This is F2 generation. We are producing F2 generation. From this, what are the gametes here? This is capital R, small r. That means here selfing means capital R, small r into capital R, small r to produce the F2. This is the F2 generation we are producing. To produce F2, from this parent, two gametes are coming. From this parent, two gametes are coming, capital R, small r. Here, this is male parent, these are female parent. Capital R, what could be there? This is the red color flowers. And capital R, small r, what colors will come? Pink color flower. And again, what would be there here? This is capital R. Now, capital R, small r, what is this flower? Pink color flower. Small r, what flowers will come? White color flowers will come. Now you see phenotypically, what is the ratio? Phenotypic, red is 1, pink is 2. Red, how many plants will be there? 1. Pink, how many plants will be there? 2. And white, how many plants will be there? 1. So what is the ratio phenotypically? 1:2:1. You come for genotype. It is homozygous head. Capital R, how many will be there? 1. Capital R, small r, how many will be there? 2. Small r, how many will be there? 1. That is homozygous dominant, heterozygous dominant, homozygous recessive. Genotypically 1:1, phenotypically 1:1. So what is the phenomenon? It is showing this phenotypic ratio 1:2:1, genotypic ratio 1:2:1. What is this here? Incomplete dominance. But in incomplete dominance, what is this? 4 plants only coming means monohybrid cross. 16 plants comes means dihybrid. So that here 1:1 and having this here same phenotypic genotypic, that is monohybrid cross with incomplete dominance. So here 11th question answer is the D answer is correct. Incomplete dominance. Snapdragon plant, here we can see this. It's question number 25. Read the following 4 statements A to D. Then, after reading these statements here, how many above statements are right? 2 statements are, 3 statements are, 4 statements are, 1 statement are, you see. Here, read the following. 4 statements. Following 4 statements A to D. In transcription, number 1, in transcription, adenosine pair with uracil. Regulation of flag operand by repressor is referred to as a regulation of flag operand by repressor is referred to as a positive regulation. Positive regulation. C. The human genome has approximately approximately 50,000 genes. Hemophilia is a sex-linked recessive disease. Hemophilia is a sex-linked recessive disease. So answer A is 2 statements, B is 3 statements, C is 4 statements, D is 1 statement. Here, of these, what we did find out here, how many of the above statements are correct? How many of the above statements correct? How many of the above statements are correct? So here you can find out this how many statements. Suppose in transcription, what is the pairing rule in transcription? C can bind with C, A can bind with U only. Here A can pair with uracil. So this is correct or wrong? A is pairing with uracil, correct one. But regulation of flag operand by repressor is positive regulation or negative regulation? Negative regulation. Repressor is activated, gene expression is inhibited. Gene expression could not done if repressor activated. So that here negative regulation, not a positive regulation. So that here this is the wrong one. The human genome has approximately how many genes are there? The estimated genes are 30,000 genes here. 30,000 genes approximately. As per human genome project here, human genome has approximately 30,000. So that here wrong. Hemophilia, what kind of disease here? Sex-linked recessive disease. Color blindness, hemophilia, sex-linked disorder. What is this sex-linked recessive? So here, how many statements? A and B correct. So answer 2 statements are correct. So in this regard here, 4 statements were given. Out of 4 here, this is here gene regulation. These all are this. What would be the transcription point? This is the translation point. It is human genome. It is the sex-linked disorders. So here, out of 4 aspects here, 2 are the A and D are correct, B and C are wrong. 2 statements correct. So answer is the what is A is correct. 2 statements correct. So next here, we go for 26. A test cross is carried out. 26. A test cross is carried out. Is carried out. What purpose we are carried out the test cross? To find out the genotype. In the what is this given test? Given the cross here, what the offsprings have been produced in this offspring's genotype. To find out the genotype of this offspring, we have to perform this here test cross. So that here, what is the actions you see? Determine the genotype of plant at F2. Predict whether two traits are linked. Assess the number of alleles of each gene. Determine whether two species or varieties will breed successfully. So answer, what is the answer here? Determine the genotype of the plant. Answer A, here you see, determine the genotype. Determine the genotype here. Determine the genotype of a plant. Of a plant at F2. Here test cross is done. What purpose here? For determination of the genotype. Now you see 27, when two unrelated individuals are crossed, the performance of a F2 hybrid is often superior to both the parents. This phenomenon is called as. Generally, two unrelated individuals are crossed. A and B is crossed. 27 question. Individual A, individual B, these two are crossed. When these two are crossed here, C has been produced. What is the A plant feature? A plant is suppose here small size fruit. Fruit size is small. B plant is big size fruit. But here there is no taste. There is no taste, big size. Size is big. But here size, small size. The size is small, but good taste is there. Sweet taste is there. Whatever is here, sweet taste. Now you see what is desirable quality. Big size is desirable quality. Next one, good taste, sweet taste. So now this is big size and sweet taste. Sweet taste, both characters came. Actually, this parent only big size, no taste. And here this parent, what is the small size but good taste. Here taste is quality character. Here size is quality. Both are seen in this. If on crossing these two parents, both characters are seen in offspring, which will be called here hybrid vigor. This is the hybrid vigor. That is also called heterosis. Heterosis. So what is the question? This question is heterosis. What is the question here? 27 question. You see, two unrelated individuals or lines are crossed. The performance of F1 hybrid is often superior to both parents. The superiority of offspring over their parents is called as the heterosis. Superiority of offspring. What is the 27? What is the superiority? Superiority of offspring is called as superiority of offspring. How is superiority of offspring here? Answer here you see heterosis. The answer is the heterosis. Superiority of parents is called heterosis. You know, you check with your served sheets. Answer here heterosis. Next 28 you see which of the following condition correctly describe the manner of determining the sex. Homozygous sex chromosome determine the female sex in parts. Correct description. We need this here 28. Question number 28. Question number 28. Correct description we need. What we needed here? Correct description. In this correct description, what is the correct description? We need option A. What is the option A? Here homozygous sex chromosome ZZ determines what sex here? Female sex. Female sex in birds. Female sex in birds. It is correct or female sex are wrong. Male sex ZZ determine female sex in bird wrong. What is their meaning? ZW determine. Next B. Option B, X0 determine male sex. X0 determine male sex in grasshopper. It is correct. Here X0 determine male sex. Correct. XX, X0 method occurs in this here grasshopper. X0 is the male, XX is female. So this is correct. Option C, you see what they have given. A X0 condition in human form, the Turner syndrome, determine female sex. X0. Here it is a Turner syndrome. Turner syndrome. X0 Turner, determine female sex. Determine female sex. It is correct, determine female sex here. Generally, it is for determination of sex. What is the methods we are using? XX, XY. In human being here, female sex here in human being, they are given. Female sex in human being. In human being, what we are using? XX, XY method. What is determining the female sex? XX, not X0. X0, XX, X0 applicable for insects, not for man. So that here XX, XY method applicable for this here man, not this X0 method. This is wrong. Next here, D statement, D. What is the statement D? One X and one Y chromosome. Chromosome XX produce male in Drosophila. Option D. XX produce male. XX produce male in case of Drosophila. XX produce male. Here XX produce female. In Drosophila, this is wrong. XX produce, what can produce XX here? Female, not male. So this is wrong, this is wrong, this is. X0 determines male sex in Drosophila, correct. So 28 is the D answer. 29, which one of the following condition of zygotic cell would lead to the birth of a normal human female child? Female child gene configuration 2X chromosomes. 29, what is the female genotype? Female genotype here. What is the female genotype in human being? In case of human being here, what is the female genotype? 2X chromosomes. Option A, 2X chromosomes will be there. XX chromosomes. How many X chromosomes will be there? 2X chromosomes. You see option here, 2X chromosome, only one Y, only one X, only X and Y. So what is the female genotype here? 2X chromosomes. So answer is A. Next year, test cross in plants or Drosophila involves crossing. What is the test cross? Double recessive parent. F1 hybrid with recessive genotype is called as the test cross. What is meant by test cross here? Question number 30. This is the 29. Question number 30. You come for question number 30. What is meant by test cross here? You have to see this test cross. What is test cross here? F1 with recessive parent. What is meant by test cross here? F1 generation. Suppose you see these are the parents, A and here A. These are the parents. If cross is made from this A sperm, it is A ova. These two are crossing, A will come. This is the F1. This F1 is mated with recessive parent, of which will be called test cross. What is meant by test cross here? F1. This is A. This is F1. F1 with recessive parent. It is dominant parent, recessive parent. So this is F1 with recessive parent is called as the test cross. The question number you have to find out test cross. What is test cross here? You see between genotypes with two genotypes with recessive trait. Not between two genotypes with recessive trait. Between two F1 hybrids. F1 hybrid with double recessive or simply recessive. If it is a dihybrid test class, double recessive. Monohybrid test class, single recessive. Whatever it is, monohybrid, dihybrid means you take AAG, BBG into AABB. At that case, what will come? AABB. This is the F1. It is double recessive. Again, what we are doing? F1 with what is this here? AABB. So here it is the F1 with double recessive parent. How it will be called? Test class. So in this case, what is the answer? The option C is correct. What is the answer? Test class answer? Option C. What is the option C? You see the F1 with double recessive parent, that is called as the test class. So this is the test class information we have discussed here. The remaining questions here.
tomorrow we can discuss. Okay, thank you one and all. Do the practice regularly, okay, until the completion of your NEET examination. Don't waste your time. Thank you.